And welcome to Module 10. We're now going to make kind of a shift in this class. Up until now, we've been talking about bacteria, how we describe them, what they do, etc., etc., etc. In the last 3 modules, we're going to talk about us a little bit. All right, we're going to talk about how do we fight bacterial and viral infection? How do we do things in order to protect ourselves from disease? And then a little bit at the end of what happens when our very fine-tuned immune system kind of goes haywire and doesn't do what it's supposed to do, right? Both the good side and the bad side of these things, right? Module 10 is all about the innate immune system, something we call the non-specific immune response. So we'll talk about that, the importance of that, and then we'll continue on with the other elements of the immune system in later modules. So with that said, let's begin. Innate immunity, or the innate immune response, is the non-specific response to infection. We'll talk a little bit about what that means exactly. When we say immunity, what we're referring to is the ability to protect against disease, not just from microbes, but also from their products. As we learned that some diseases are caused not by the presence of the pathogen, but the toxins and other things that are produced by the pathogen. Sometimes we'll use the term resistance to also refer to immunity. The opposite of that is susceptibility, right? Which is a vulnerability or a lack of immunity. And for the most part, we all are susceptible to most diseases out there. And then over the course of time, we can either gain immunity to diseases, whether it's through active infection or whether it's through vaccination and immunization, which we'll talk about next module, I think. Yeah, this idea of memory. 2 general arms of the immune system, the innate or non-specific immune system, which we'll talk about in this module, and then the adaptive or specific immune system, which we'll talk about in the next module. We often talk about these as being 2 separate arms of the immune system, but the reality is that they are very much interconnected, right? So a lot of what happens in the innate immune response kind of talks to the adaptive immune response, and the adaptive immune response is very much dependent on the innate immune response. So they're very much hand in hand, okay? So let's talk about the innate immunity. What do we mean by non-specific immunity, right? Here we're talking about first line of defense, right? So, I always like to imagine that our body is like a castle, right? We're like this fortress. And then so the innate immune response are the many levels, the barriers that an invader has to overcome in order to breach or get access to this castle. They are what we call non-specific in that they don't target any specific pathogen, right? So they do. Where are we? There we are. They do not target any specific pathogen, right? So the way the system works, again, think of like a moat. Moat doesn't specifically say, oh, we're gonna defend ourselves against horses or archers or all these things, right? This is like, we're gonna have a moat, and then if you gotta scale our wall, you gotta deal with this moat, right? So that's the general idea here with this non-specific immune system, right? Same idea. Just we're gonna have all these mechanisms, these barriers, these things that a pathogen has to overcome, right? Not one specific pathogen. Of course, each element of our non-specific immune system does deal with various types of pathogens, as we'll talk about, right? But the response is always going to be the same, right? It's innate. They're built in. They're just there. So it's something, again, that the pathogen invades, and then our body says, okay, let's just do this. And then you're just going to have whatever mechanisms that are there, always going to happen the exact same time. And then we'll contrast this next module with the adaptive immune system. There is no memory element. So if you beat a pathogen one time, you got to do the exact same thing to beat the pathogen the second time, which is different from the adaptive immune response, which is, it reacts differently next time it sees the same pathogen. And the innate immune response is very rapid, right? Almost immediate, right? And we'll talk about some elements that, if you think about it, you will definitely see the immediacy of the innate immune response, right? Whereas the adaptive immune response takes a lot longer, right? And again, very much playing in conjunction with each other, right? Both arms of the immune system are heavily dependent on each other. 3 main components of the innate immune response. We have our physical defenses, chemical defenses, cellular defenses. And so we'll talk about each of these. And of course, each of these are going to have parts of these, okay? So with respect to physical defenses, microbes have to overcome them when they invade. So now you imagine a microbe is going to come and try to attack you. And then in order to get at the good part, the places the microbes want to be in, it has to get and overcome these defenses, right? So there are three general types of physical defenses. We got our barriers, we have mechanical defenses, and then we have our microbiome, right? And these things are all elements that we are quite familiar with because, you know, we all have them, and we hopefully are aware of how these things work, right? But physical barriers are essentially barriers, things that like walls, like moats and things like that, that a pathogen has to be able to breach, right? And oftentimes they're breached when there are breaks in these barriers. The mechanical defenses are mechanisms to remove pathogens. So things that are sweeping, things that are doing that kind of stuff to get rid of pathogens. And the microbiome are the bacteria and the fungi that live with us that essentially compete with pathogens. So we'll talk about each of these elements quite briefly. So with respect to physical barriers, again, the idea is that it just prevents microbes from getting to the sensitive tissues, right? And the most important barrier is the skin, and I guess the barriers are the mucous membrane. So these are the things that are, these barriers are the most important with respect to access and preventing microbes from blocking or from getting to our sensitive tissues, right? So we see our skin, it's this large covering, right? We say it's the largest organ in your body because it covers the whole body, right? And there are many things that we can do to prevent microbes from getting at it, right? So first of all, the skin is extremely tight, right? These things are called cell junctions. This would be one cell, this would be another cell. And in these cell junctions, they're like glued together essentially, right? To prevent microbes from squeezing. So if a microbe wants to get at the soft tissue, it has to figure out how can we break these junctions here, right? These tight junctions or these gap junctions, and be able to break apart these barriers, allowing microbes access to the middle. Okay, so let's talk a little bit more about the skin. It's a first barrier. There are three layers: the epidermis, the dermis, and the hypodermis. The hypodermis is kind of the last part right before all the sensitive stuff. So this is kind of where microbes want to get access to, right? Once they get access to the hypodermis, things start to be a little bit more problematic, right? Now, you know, we think of our skin as just this flat surface, right? But this is this cross section of the skin. So first of all, this is what we see, right? And so even here, you know, it's kind of this this vast, almost like a desert environment, right? We produce these fatty acids that keep the environment very dry and a little bit acidic, right? So our skin's a bit acidic. It inhibits microbes from growing. It's dry for the most part, right? Unless we're sweating, right? So it doesn't provide a nice environment for microbes to be able to grow, right? So the skin level itself is the epidermis. It's just not a nice place for microbes to be, right? So they need to get access underneath. I think we all learned this before, right? But the cells on top, we have this layer of dead cells right on top here, right? And they're shed, so they're constantly shed. So if a bacterium, evil pathogen, happens to land right there, right? Eventually that skin cell is going to be shed, and then the bacterium floats away with it, right? And then we're constantly shed the top layer, and then we make new skin cells underneath, right? So it's this idea of constantly replacing and removing stuff that's on top, and then we can get brand new skin cells underneath, right? So when you have a pathogen, typically it's breaks or the parenteral route that allows microbes to get access. So when you break this thing and then you get it into the softer layers underneath, that's when microbes cause problems, right? But the skin itself is quite a formidable zone for the microbe to get past. Similarly, the mucous membranes are they cover these membranes that cover most of our tracks, our kind of pathways that exist the outside, right? So we have our membranes in our eyes that are a type of mucous membrane. Most famously, we have our respiratory tract, our intestinal tract, our urinary tract. All of these things are coated in mucous membranes, and what mucous membranes do is they secrete mucus. Notice the spelling, by the way, right? Mucous membrane is a proper noun, so we use the British spelling, but the stuff that it produces is mucus, which is our American spelling, right? And mucus, as we know, as we've seen, is this sticky substance that's constantly secreted by the mucous membranes, right? And then this protects the cells by essentially trapping all the debris. So all the debris, all the things get stuck in this mucus, right? And if it gets full and has a lot of debris in it, we simply cough it up, we sneeze it out, or we swallow it, right? And so that's how we can get rid of most of the debris. And then we know that when we have respiratory infections, right, or when you inhale a bunch of dust or something like that, the immediate response is that you get this mucus secretion. It's all like, oh no, things are coming in. And then it starts to secrete this mucus and trap it. And then you start coughing and sneezing and all this kind of stuff to just forcibly expel things out, right? So these barriers are things that they have to get by. There are mechanisms, and that's kind of a mechanism to removal. We'll talk more about it a little bit later. Or not, called a mucociliary escalator, right? And what that means is that we have the ciliated epithelial cells in our upper respiratory tract that are constantly moving, right? So here we would have microbes stuck inside the little cilia, that's these little hairs, and they're constantly sweeping up, right? You know, imagine like these little algae, these are the ocean plants, right? And then this constant like current, so to speak, right? And then they kind of pass the microbe up, and then eventually out, right? So as we get it away from the lungs, we then, again, at some point, we'll swallow it, we'll cough it out, we'll sneeze it out, blow our nose, whatever the case may be, right? And they help protect the lower respiratory tract, which are very sensitive, right? Once you start getting stuff in your lower respiratory tract, there are big issues because that's where oxygen exchange happens. That's where these things, you know, all the good stuff happens, right? So if that stuff fills with mucus, all of a sudden now it becomes much harder to breathe and all that kind of stuff. So the mucociliary escalator, very important in the upper respiratory tract, yeah? And then, you know, we know the importance of this because things like smoking inhibit the mucociliary escalator, so the sweeping motion is not quite as vigorous. Something like cystic fibrosis is this genetic disease when the mucus produced is extremely thick, and then this super thick mucus inhibits this sweeping action, right? And then so people who smoke, people who have cystic fibrosis are very prone to respiratory infections because the action of that mucociliary escalator has been inhibited, right? So it doesn't work quite so well, so they're more prone to respiratory tract infections. The intestinal tract has a very similar mechanism, right? The idea of sweeping, in this case it's called peristalsis, where the muscles of the lining start to push things out and then just move things away, right? So moves mucus out and ultimately is excreted in the feces. In this case, the movement will always be, you know, comes in one way and then out the other way. But similar idea, things get stuck in the mucus and then they get pushed out, excreted in their feces. And finally, the last physical barrier we'll talk about is the endothelia. And these are the cells that line many tissues, right? So if our microbes, if pathogens are able to get access to the internal parts, right? All our organs, all the good stuff are still wrapped in tissues, and these are called the endothelia, right? It's almost, think of it like the skin for your organs, right? So all your organs, your urinary tract, your blood vessels, all of these things have these very, this coating very tightly packed together and prevents pathogens from invading into tissues, right? So, you know, this blood-brain barrier, you know, whereas blood circulates and then the little things come through, the endothelia and the blood-brain barrier prevents things from big things like microbes, like viruses, from squeezing out into the central nervous system and causing some big-time issues. All right, so the commonality here now are these barriers, right? So microbes come in, they have to get past these things. If they land, and then again, we think of them as separate, or we talk about them as separate, but they're all joined together. We have these mechanical defenses then that physically remove pathogens, right? So we talked about the mucociliary escalator, right? This idea where things are being swept away, and then stuff like coughing and sneezing, right? Those will be mechanical defenses. They help, you know, things that are trapped within the membranes, right? The mucous membrane. So the mucus will be the physical barrier, right? the mechanical defense would be the sweeping and then the coughing and the sneezing, right? So these are forceful mechanisms to get out, right? Anytime you get anything in your eye, what's the immediate response? You're going to start tearing up, right? And the tearing up is not because of the pain, right? But because the tears help, once again, wash away. And then you start blinking, and the blinking starts washing away any debris in your eyes, right? The eyelids help trap stuff, constant flushing, using tears, using urine. All these things are extremely powerful mechanical defenses to help physically remove pathogens. Okay? So imagine now microbes comes in. It's able to get past the initial physical barriers. It gets stuck in the mucus, but it's able to get past that because there's just so many of them that many of them get through. It's able to get past all the sweeping and all this kind of stuff, and then they get at the good part, right? So now we have the pathogen is now invaded and starting to try to occupy the inside of your body, right? Well, it turns out that as we talked about way at the beginning of the semester, we have microbes that live within us, right? Our microbiome, the resident microbes that live within us, right? And I think we talked about how it's been estimated that we are 10 billion human cells, 10 billion, 1 billion. And then we have like about 100 billion microbes associated in and around us, right? So, we have all these microbes that live there, right? And so the idea here is that the microbes, they help through this phenomenon called competitive exclusion, right? So they compete with the incoming pathogens, and they exclude them, right, by preventing them from getting a foothold, right? And generally that our microbiome or our resident microbes are very good at hanging inside our body, better than the pathogens, right? So we mentioned Clostridium difficile, right? And how Clostridium difficile can infect a depleted GI tract. Typically, do you guys remember how that happens? How does one get a depleted GI tract, right? Yeah, through antibiotic treatment, right? So when you're taking your antibiotics and the microbes start to die off, right? Our resident microbes, they start to die off. Clostridium difficile can now grab a spot in the intestinal tract and then cause extremely potent, very deadly GI illness, right? So much so that the best treatment for this is not antibiotics, because remember, antibiotics is the reason that we're there in the first place. It's through fecal transplant therapy, and it is exactly like what it sounds like, right? So you eat these capsules that are filled with poop of a healthy person. I know, I'm not making this up, right? You literally eat these capsules that are filled with the poop of a healthy person. And the idea is that this healthy person has microbes that are very good and at keeping bad guys out. And then they're able to then outcompete the Clostridium difficile, right? And this is, so, it sounds nuts, right? But the success rate of this is something like 80, 90% or something like that. 90, I forget, some really high success rate percentage, right? And it's this, there are very few therapies that have such a high and such a fast success rate, right? But fecal therapy right now it's the thing, and you can imagine in the future that we will have figured out what microbes are the most important, and you can get it in powder form, so then you don't have to literally eat someone's poop to get healthy, right? But nowadays that's what we're doing. You're eating someone's poop to get healthy, right? But the Clostridium difficile is so bad that you are willing to do it. The other thing our resident microbes do is that they compete for nutrients, right? So many yeast infections, which are not quite as fast growing as bacteria, they often get outcompeted because they just can't eat as fast as the bacteria that are living there, right? So they make some kind of an overall less able to compete. And then we'll talk later about some of the chemical defenses. Some of the chemical defenses produced by our body are actually produced by our resident microbes, right? So again, it's a very tough environment, very tough thing for microbes to deal with. Pathogens have to come in, they got to get past the barriers, they got to, prevent being swept away from the tears and all that kind of stuff, and then they got to deal with all these microbes that are hanging out there, right? So a quick overview. This is in your book of the physical defenses. You can pause here and then do a quick overview if you want to. we're going to move in and talk about the chemical defenses, right? So what are these things that are in our body? And then these are what we call chemical, and the key word here is mediators, right? So they mediate are involved in our immune response, right? So the definition is quite vague, right? Widely diverse chemicals found in fluids and tissues. We have tons and tons and tons of mediators. We'll talk about mediators in so many different forms, so many times that you'll probably get tired of me saying the word mediators, right? And then there are two general types of mediators based on where they come from: endogenous and exogenous. Endo referring to in, exo from out, right? So endogenous mediators are produced by us, by our body cells. Exogenous mediators are produced by microbes, right? So some of the mediators, see, I told you, you get sick of me saying the word mediators because I am already. Some of the mediators are produced by the microbes, and they help, once again, in this idea of outcompeting the microbes. Some of them are ever present, so they're always going to be there. Some of them are produced in response to pathogens' presence, and we'll see some of examples of this as we go along. So let's talk about some of these mediators, right? So these, and again, they exist in many places. We'll talk about the most important ones are the ones in our body fluids, right? Here we have both endogenous and exogenous mediators. And the one that's probably the most recognizable in our skin is called sebum, right? Sebum is this oily substance, right? And the idea behind sebum, and it's produced by in our pores, is that it blocks, right? So this oily substance, so it prevents access to the good stuff at the bottom, right, down here, by simply blocking these pores, right? And then we know sometimes that causes us issues because if you produce too much sebum and it doesn't get cleared away, right? It's supposed to like be produced, block, and then get swept out, and that's why our skin can be slightly oily, which is, again, is kind of this physical response, right? But this blocks the pores, and then we know sometimes the pores get plugged and we get these yucky pimples and blackheads and all this kind of stuff. The irritating thing is some microbes have figured out how to eat it. It's sort of like, hey, we're going to use this to protect ourselves. And the microbes are like, I don't care, I like it, yum, 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 right? So they eat it up, right? And then so what happens then is that as they eat the sebum, the metabolic byproducts then start to really block this thing, and then leading to stuff like acne, right? So it turns out that we actually produce more sebum during parts of our lives. It's hormonally regulated, right? So during adolescence and puberty, you understand, right? During puberty, we tend to overproduce sebum, right? As the hormones start to really kick out, and with puberty, we start to overproduce sebum, right? And then this then allows some of the microbes, and some of them are just already hanging on our skin, to just really eat this stuff up. And then so that's where some of our acne problems come from, right? And the reason that it happens during puberty is because of this fact that we overproduce it, right? So if you are currently cursing your skin for all this acne that you may have right now, or just kind of the remnants of as you survived high school, the good news is that it'll calm down, right? You will produce less sebum, and then acne will become less of a problem, I promise you, hopefully. There are other mediators, right? And so we have antibacterial enzymes, so lactoperoxidase, lysozyme, that's probably our most famous one. These are found in mucus and tears, and almost every single fluid that we have lysozyme, right? We have stomach acid, right? So the acidity in our stomach kills bacteria. And then within that, as it gets past the stomach into the small intestines, we have enzymes that degrade peptides or proteins and lipids and carbohydrates. We have peptides, as we'll talk about. We have bile that create this very inhospitable environment, right? That help eliminate a lot of these pathogens. Many of our urinary tract, our urinary tract and our vaginal fluids are quite acidic, once again inhibiting the growth of a lot of microbes, and even our earwax is acidic, right? They're very rich in fatty acids, right? So all these things are these various chemical environments that do things like lower pH, degrade enzymes, or sorry, degrade proteins via enzymes, antibacterial activity, all these types of things exist, once again, making it very difficult for a pathogen to come and invade. Another interesting chemical defense are these things called antimicrobial peptides, or AMPs. These are broad spectrum antimicrobials. A peptide, if you recall, is just a small protein. So we're talking anywhere from like 12 to, let's go 25-ish amino acids. Okay, so very small little proteins. Very diverse in mechanism, right? And sometimes the mechanism isn't even clearly understood. We know that they do things like damage membranes. They can interfere with cell walls. They can destroy RNA, DNA, right? Most of them are endogenous, so we produce these things. Some are exogenous, so some of these peptides are produced by microbes that kind of live with us, and they're very interesting, right? They were initially characterized in some invertebrates, and it turns out it's a very rudimentary immune system, right? So in invertebrates which don't have an actual immune response, they do produce these peptides, and peptides are very small. We can actually synthesize them quite easily in the lab, and these are an interesting avenue for next generation antimicrobials, right? Because we can synthesize them, and because they're so small, we can modify them quite readily. And to be honest, we can make rando 15 amino acid peptides, and then just let's see what happens, right? So the list down here is not something I want you to memorize. Just understand that they are there. So we have bacteriocins, defensins. We're going to talk about defensins again very quickly with respect to things like macrophages and neutrophils. So put that in your mind bank. Defensins, macrophages, neutrophils. But for the most part, we are able to have many things. Bacteriocins are endogenous. They are produced by resident microbes. And then we have mediators in the blood, and we'll talk more about these later, right? So plasma is simply the fluid portion of the blood, and there are many immune mediators. We will talk more about these. No, we'll talk about them right now. So we have three general classes: acute phase proteins, complement proteins, cytokines. Just again, surface. We'll talk a little bit about them. So let's mention the acute phase proteins. These are all something to do in the inflammatory response. And we'll learn later that the inflammatory response is acute in that it's very rapid, very short-lived. These are primarily produced in the liver. And again, the function of these acute phase proteins, such a broad term, right? But basically acute phase means that these are proteins that are produced immediately in response to inflammation, all right? And they do many things like, we'll learn later what this term means, opsonization, right? Here we'll talk later about how some of them can sequester iron, which is a very important nutrient for pathogens. You can hear that it's forming blood clots, activates complement, which we're going to talk about in our next slide, right? You don't have to memorize this. Just understand that acute phase proteins produce an inflammatory response, right? And then they have multiple functions, okay? So let's talk about complement then, right? So complement is a really neat, kind of complex system. There's a group of 30 plus plasma proteins, part of our innate defense system. And again, it's a very rudimentary immune system, right? There are lower vertebrates and invertebrates, I believe. Ooh, don't quote me on that one. That have complement systems that help them protect against infection. And then the important part of the complement system is that it helps fight infection immediately, i.e. part of our innate immune response, but there are elements that connect it to the adaptive immunity as well. So once again, this bridge between our innate and adaptive immune response. Basically, they circulate their blood. I think there are 9 proteins that just kind of circulate until they are quote unquote activated, right? And there are 3 ways to activate them. I don't want to talk about them, but they're called the classical pathway, lectin pathway, alternative pathway. Activation can be a little on the complex side, so we're just gonna know that there are different ways complement is activated, and even later we'll say, oh yeah, this is one of the ways to activate complement, but it doesn't matter. And then once complement gets activated, there are 4 general things that happens, right? One is called opsonization. I promise you we talk about this again, right? And what opsonization is that it's the coating of a pathogen, right? So it's called the opsonin. So these things are called opsonins, right? and they just coat the pathogen, and then it allows our white blood cells to grab them much easier, right? Because some microbes are able to kind of hide from the immune system, right? So by coating them with opsonins, all of a sudden now our white blood cells, they don't have to recognize the pathogen, they just recognize the opsonin. They grab it and then they eat it. Another outcome is inflammation. So it's one of the ways that inflammation gets triggered, right? And we'll talk about inflammation a little bit later. Chemotaxis is a very important thing. Chemotaxis, we talked about long time ago, right? Is this attraction, or chemotaxis means the ability to attract or repel cells according to using a chemical. In this case, we're going to use chemicals to bring white blood cells, right? So here with chemotaxis, we're referencing the ability of bringing white blood cells to the site of infection. And then finally, cytolysis. Remember, lysis means to break, cyto means cell. So here we're talking about the destruction of a pathogen by these things called membrane attack complex, which are essentially just big holes in the membrane, right? So here, this is kind of what cytolysis is. Big holes in the membrane, and then everything leaks out, and then the pathogen dies, right? So 3 ways to activate complement, and then 4 outcomes. Opsonization, which allows white blood cells to grab and eat them. Inflammation, we'll talk about inflammation a little bit later. Chemotaxis brings white blood cells to site of infection. And then cytolysis, physical killing of the microbe by essentially making holes in the membrane. Okay, the last plasma contained, the last plasma protein mediator is the cytokine, right? Now, cytokine is simply communication between cells, or communication signal between cells. And we're gonna use the word cytokine so much. Cytokine, right? But basically, it's all cells can secrete cytokines, all cells can receive cytokines, right? So many different kinds of cytokines, so many different outcomes. 3 general types I'd like us to know. Autocrine means that, auto is kind of like, think automatic, right? It means the cell will send the signal for itself, right? It's sort of like, haha, I'm turning myself or activating myself using these things, right? Paracrine is a signal to a nearby cell, so it's sort of like cell gets activated, sends out these cytokines with this idea that all my neighboring cells are going to get activated, right? It's a signal for my neighbors. And an endocrine is a signal for things far away, right? So it goes to the bloodstream and it goes to a very far away cell, right? So 3 general types of molecular messengers, but the overall thing with the cytokine is it's secreted by a cell to tell other cells what to do, okay? We are going to run into many types of cytokines, right? Again, the level of cytokine is quite complex, so we're just going to keep it, like always, kind of surface, right? 3, with respect to the immune system, because there are so many different cytokines, with respect to the immune system, there are 3 classes that we will talk about. The interleukins, they're called interleukins. Later we'll learn leukocyte refers to white blood cells. They used to be thought that they're secreted by white blood cells for white blood cells. It turns out that almost all cells can secrete an interleukin. Almost all cells can receive signals from interleukins, right? But basically, the function of the interleukin is to modulate aspects of the immune system. I know, so vague, right? So many different aspects, right? But there are things that it can do to activate, shut down, tell other immune system cells or immune response what to do, right? Chemokines are cytokines that attract white blood cells. This whole idea of chemotaxis again, right? So chemokines are cytokines produced by cells that bring white blood cells to site of infection. And then there are interferons, and interferons are defense against viruses, right? So what they do is that they interfere with viruses. So 2 types of interferons, type 1 and type 2. Type 1 interferons inhibit viral replication, or they destroy viral infected cells, right? So here, for example, we got this virus. This cell is infected. Boom. It sends out a cytokine to its neighboring cells, right? What kind of cytokine would this be? Autocrine, paracrine, or endocrine? These would be paracrine, right? Because it's talking to its neighboring cells. And a couple of things can happen. the cytokine can, or this interferon, type 1 interferon, can tell this cell, hey man, you know what? You're going to get infected by a virus, so why don't you destroy any RNA that's kicking around you, or why don't you just reduce just like how much protein you make, right? So all of a sudden now, this virus is essentially protected from virus attack because it has stopped much of its metabolic activity with respect to RNA and protein synthesis, which is what the virus requires. So the virus just kind of hangs out here, says, oh, poo, nothing's going on, and then it just kind of eventually gets destroyed, right? If the virus has already infected its neighboring cell, this cytokine now says, okay, you're infected, let's, we're gonna kill you, all right? So signal that says, hey cell, you're infected, you know what to do, and the cell essentially commits suicide, all right? And we'll talk a little bit about what apoptosis is, and then the type 2 interferons activate immune cells, again, with respect to viral infection, right? So type 1 inhibit replication and destroy infected cells, right? So they are telling cells how to deal with the virus that's around, and type 2 interferons activate immune cells. So notice that, by the way, interferons don't actually deal with the virus. They don't bind to the virus. They don't eliminate the virus, right? They do it by telling other cells what to do, because cytokines, cell-to-cell signaling. Finally, the last mediator we'll talk about, mediators. We'll talk about our inflammation mediators. It's just a way to trigger inflammation and fever, which later we'll talk about, non-specific immune response, and we'll see the importance of inflammation and fever. Let's just mention a little bit about some of the inflammation mediators, which some of these we'll see again, right? So histamine is probably the most important. It triggers a bronchoconstriction, broncho referring to our respiratory tract, right? So it tells our the tract in our lungs and our respiratory pathways to get nice and tight, right? And there are many reasons that we want to do that kind of stuff, right? But that's why sometimes, you know, during inflammation, you have a little bit of trouble breathing because of this whole constriction, again, preventing pathogens from getting at you. Leukotrienes are a pro-inflammatory lipid, right? And so the fact that it's a lipid means that it kicks around a little bit longer, right? They don't get degraded quite as fast, right? So overall, the response from leukotrienes are longer lasting. They're more potent. Prostaglandins are pro-inflammatory and fever-inducing, right? So they help with the fever response as well. And then bradykinins cause edema, which is swelling. And we'll kind of see some of the responses or the outcomes of some of these just a little bit later. Okay, that's it for this section. Now's a good time to take a little bit of a break, right? If you don't feel like taking a break, let's continue and talk about our cellular defenses, right? So the cellular defenses are all about the cell. So we're going to go through a list of cells. So we previously mentioned that plasma was the fluid portion of blood. The non-fluid portion of blood is called the formed elements. And we call them that because all the formed elements are formed from the same stem cells found in bone marrow. And we'll talk about that in just a second. Three categories of formed elements: red blood cells, white blood cells, and platelets. Red blood cells are called erythrocytes. White blood cells are called leukocytes, and they're called that because cytes referring to cells. Erythro means red, leuko means white. Very clever, right? And then platelets are called thrombocytes. Thrombo referring to clotting. Oh, shoot. I'm pretty sure that's what that means, right? So, but sometimes you're going to see the terms interchangeably. Okay, usually I like to say red, white, and platelets, but sometimes you're going to see, especially with respect to conditions, erythrocytes, leukocytes, thrombocytes. I don't want you to memorize this. I just want you to understand where they come from, right? So there's a multipotent hematopoietic stem cell, right? We call it multipotent because this one stem cell can become many different cells. A little different from pluripotent. Pluripotent means this cell can become any cell, right? But here, this cell can become any of our blood cells. It's a hematopoietic stem cell, right? Referring to blood and stuff. And so it's this one here then after a number of signals, and it's kind of like every time you make a choice, you can't go back, right? So we have these myeloid stem cells, lymphoid stem cells, and they all become these various formed elements that we will talk about, right? But all the formed elements in blood all come from this one stem cell right there, this multipotent stem cell that lives in the bone marrow. Okay, so don't memorize this chart, but we will talk about pretty much every single one of these cells. And then same with this. Don't memorize this, but that's definitely, you know, in terms of interest, right? This is a nice summary of function, what they look like, and then even some interesting little fun facts, right? So if you want to pause, it's actually in your book, but if you want to pause here and just read it, really nice things to know, especially if you're going to move on to the clinical field, right? Notice the red blood cells are the most abundant, right? Because of course it gives us the primary function in our blood, right? And then we have a number of different white blood cells, of which we will talk about all of them. All of them. Okay, so with that said, let's move on to the white blood cells, right? And there are two general types of white blood cells called granulocytes. Oh, I guess we're going to go back here. Called granulocytes and agranulocytes. Okay, and so we'll talk about these, and then we have some that are kind of in between, as we will mention. So granulocytes are leukocytes. They contain granules, right, that are under the microscope. When you look at them, say, oh, we see these are the granules. And it's the granules that form function. So the granules are actually membrane enclosed sacs that contain some sort of mediator. There's that word again. All right, and it's this mediator then that provides function, you know, in part to these granulocytes. Three types: neutrophils, eosinophils, and basophils. So let's start with neutrophils, probably the most important of the granulocytes. It's the most abundant leukocyte. It's the most abundant formed element outside of red blood cells, I think, if we don't count platelets. But I'm just going to say that, and you can go fact check me later if you want to. They're also called polymorphonuclear neutrophils, or PMNs. So sometimes you're going to see that word PMN because under the microscope there are multiple, these little has multiple nuclei when you stain them, right? And then the granules we're talking about, these little things in here, right? These granules. And then the primary function of a neutrophil is to eliminate and destroy bacteria. We say extracellular because they can't do anything against bacteria that are hiding inside cells, right? But their number one job, destroy bacteria. So I want you to know that. Destroy bacteria, okay? They do this by producing defensins. Oh, there's that peptide we talked about, right? And various hydrolytic enzymes, and we'll talk about a little bit about that process. They also form this thing called a neutrophil extracellular trap, or nets, which contain a bunch of chemicals and enzymes and stuff like that, as it kind of secretes it out and helps trap microbes and then helps destroy them from the outside as well, right? So they're very powerful at eliminating bacteria. Very importantly, they can move from blood vessels into the tissue, right? So they get recruited through these various chemoattractants. They move into the tissue, and then they can fight microbes out in the tissue where usually the good stuff is going on anyways, right? As the neutrophils are starting to destroy microbes, they will die, our tissue cells die, bacteria die, all these things accumulate at the site of infection, and then that mass of fluid that accumulates is called pus, right? So whenever you see pus, it's this active fighting, this inflammation response, and typically neutrophils are going to be involved in creating this havoc, right? This pus that's formed. Eosinophils, their number one role is to protect against protozoa and helminths, right? So that's what we're going to think of them as. They also play a role in allergic reactions, okay? But primarily protect against protozoa and helminths. So that's kind of what eosinophils are known for. The granules contain histamine, which is an inflammation mediator, and this thing called major basic proteins, or MBP. All right, so histamine is how it's going to be involved in the allergic response. It's pro-inflammatory, right? The MBP binds to the surface of parasites, so it sticks to their membranes, and then it disrupts their membranes. So that's essentially how it kills the helminths, which are multicellular, or the protozoa. So it's just the eosinophils surround these protozoa or helminths, and then just injects or attacks it with these MBPs, and then starts to pop membranes from the outside. Okay, but eosinophils attack protozoa and helminths using MBPs, and can play a role in allergic reaction using histamine. And then finally, basophils or basophils, they are very important in allergic reaction, right? So these are probably the prime, along with the next cell, mediators of the immune response, and their granules contain histamine, as we just talked about, that are able to, again, trigger an inflammation response. They also contain various mediators that can recruit white blood cells. They can open up gaps between the cells, and we'll talk about the function of a role of all this in the immune response, right? And then they can be activated by both complement, which is innate immune system response, and antibodies, which is an adaptive immune response, okay? Related to basophils, very important in the allergic reaction, very well known for their ability to release histamine. Related to that are mast cells. So mast cells are very similar to basophils. They trigger inflammation. So they have many mediators that can trigger inflammation. The other neat thing that they can do is they can leave the bloodstream, move into tissues. Okay, so mast cells are kind of related to basophils. Okay, our agranulocytes are called that because they lack granules. And there are two general kinds, the lymphocytes and the monocytes. The lymphocytes include the natural killer cells and then B cells and T cells. The natural killer cells are what we're going to talk about because they're part of the innate immune response. B cells and T cells are part of the adaptive immune response, and we're going to talk all about them in module 11. Right? And then we have the monocytes. So lymphocytes and monocytes. Let's talk about natural killer cells, which is a lymphocyte. And they're called natural killer cells because they destroy cells, right? However, they don't destroy bacteria. They destroy what we call abnormal cells, right? It's a non-specific mechanism. It's extremely complex, but we're going to talk about it very simple, right? So what is an abnormal cell, right? It's, examples of this would be cancer cells, viral infected cells, right? So you say, oh, yeah, that's good stuff. I want our natural killer cells to get rid of these things, right? The simplest way to describe how we can define what an abnormal cell is through this thing here called MHC, right? And there's MHC 1 and 2. Let's just call it MHC. The long name for this is the major histocompatibility complex, MHC. And in short, they are ways that we identify our cells as self. So we all produce our unique MHC, have various molecules attached to them, right? And then so my cells all have the same MHC that says, this is me. All my cells have the same MHC. So when we have our immune system cells walking around, they'll bump into cells all the time and say, hey, show me your ID. Okay, you're a self, you're good. And they let them go, right? What happens is that when cells become infected, let's say by a virus, they stop presenting MHC because the virus infected cell is so busy making virus, it doesn't do its normal upkeep. It did not renew its identification, right? So now we have our natural killer cells coming out saying, hey, show me your MHC, show me your ID. And then they say, I left it at home. Right? Say, nope, that doesn't work for me. So then elimination, right? Any cell that does not express proper MHC gets eliminated. Okay, and it does this via something called apoptosis, which we kind of mentioned in one of our earlier slides, right? It's this controlled cell death, right? Some call it the cell death that's induced. Some call it a cell suicide because the cell says, okay, I am now doomed to make viruses for the rest of my life. So the way to help everyone is I need to get rid of myself. And it's kind of like this implosion, right? So here, I don't really like this picture so much. Really what happens is that cell starts to shrink in on itself and it becomes like this very remnant basal body, right? And so what it doesn't do is it's not an explosion. Cytolysis is an explosion, right? So apoptosis is a very controlled, no major cell debris. The important thing is that it doesn't further trigger the immune system or release these viruses that are stuck inside, right? So very important. Later we'll see MHC is very important as well with activating the immune system, the adaptive immune response, and tissue rejection. This idea of self, right? Remember MHC, self. So that's what our natural killer cells do. The lymphocyte involved in our innate immune response destroys abnormal cells, does that by recognizing its MHCs, right? The identification. Monocytes are the other agranulocyte we talked about, right? They circulate as monocytes and then they leave the blood to fight infection. When they do that, they differentiate into either macrophages or dendritic cells, okay? And all the cells are highly phagocytic, right? Phagocytic, and then simply, we'll talk about what phagocytosis is in our next slide, right? But they are capable of eating bacteria, eating cells, eating cell debris. So overall, they're a very important promoter of the immune response. They're very involved in not only clearing infection, but also triggering the adaptive immune response, as we will learn later. So remember that, okay? Macrophages, dendritic cells, very important in bridging this innate immune response to our adaptive immune response, okay? But I do want to talk about phagocytosis real quickly, right? So we have found our pathogen. This pathogen is now invaded. It's here. What are we going to do, right? So bang, we're going to eat it. Right, so phagocytosis is the eating of cells by cells, right? And actually, you know, for us, generally we talk about it, the eating of bacteria by our white blood cells, but it can also be eating dead cells, eating cells that are doomed to die, eating exploded cells and all that cell debris. All of these things are involved in phagocytosis. And the cells that are capable of phagocytosis are called phagocytes, right? So, and it's, again, very complex process that involves many things, and how these things happen is super complex. We're going to keep it nice and simple. Okay, so at its simplest, the phagocyte has to find the culprit, the thing it's supposed to eat. It has to recognize it as something that it needs to eat, and then it has to eat it. Okay, so let's talk about these processes, and some of these are actually going to sound similar to later when we talk about inflammation, right? But most of our white blood cells circulate in the blood, right? So here's our bloodstream, and then our two, you know, probably our two most important phagocytes are neutrophils and monocytes. And remember, monocytes will eventually become macrophages and dendritic cells, right? However, when you get an infection, so here we have a cut, oh, and then the bacteria came in on the cut, oh no, right? We need to get rid of these guys. And so it's our, these white blood cells have to leave the white blood cell, the bloodstream, sorry, and then get into the tissue to be able to attack them, right? So this process is called extravasation or diapedesis. I like diapedesis. That's more fun to say, right? And this gets triggered when an infected or infected cells or injured cells secrete cytokines. Cytokines, right? This mediator. So in here are these cytokines, and what happens is that the gap junctions here start to become smaller. So they make these big old holes, and then it's actually a little bit more complicated than that. I think your book probably uses way too many words to talk about it, so we won't talk about it, okay? There are resident macrophages, by the way, that are always kind of hanging out. So even though macrophages come from monocytes, and we say that they become macrophages after activation, all our tissues have these macrophages that just kind of hang out. They kind of got there and they stay there. So they are also capable of secreting the cytokines that tell, it's like, hey buddies, come over, right? So two major responses. One is to make the blood vessels more leaky, right, to allow things to squeeze through. And the other thing are these chemoattractants, right, to these chemokines that allow that attract white blood cells to the site, right? So two different types of cytokines. One, make our blood vessels more leaky. The other is to recruit or bring white blood cells to the area, right? So diapedesis or extravasation, movement of blood cells, movement of our white blood cells from our blood into our tissue, right? And they do that by squeezing through these cell junctions. Notice this only happens in the capillaries, not the veins and arteries. Veins and arteries are kind of big, right? And then if you start making those things super leaky, you're gonna have like a huge problem on your hands, right? So it actually only happens in the capillaries. So they squeeze to those tiny little vessels, right? And then they gotta make their way to that site of infection. The very important aspect of chemotaxis, right? Neutrophils come first, usually within hours of infection, because they are super abundant, super mobile, and they're ready to go. Monocytes come later. It takes them a little bit longer because there's less of them. And remember, they are monocytes, but they're more useful when they become macrophages and dendritic cells. So they actually have to differentiate as well, right? So for them, the response comes a little bit later. And so they're usually more involved in the cleanup of things, right? But when neutrophils are there, they start attacking. So they release these cytotoxic chemicals, right? We talked about the NETs, right? The neutrophil extracellular traps. So they start releasing these chemicals, these defensins. They start eating and phagocytizing bacteria, right? And start just really wreaking havoc against the bacteria that are present. So if the neutrophils do a proper job, if there aren't too many bacteria around, the neutrophils should be able to handle most of it, right? The monocytes that follow become macrophages. And then what the macrophages do is they help clean up any remaining bacteria, because it's such a very taxing process, very energy costly, right? So the neutrophils, they start to die. Many of them will die during this way. It's war. It's literally a battle, right? So many neutrophils die along the way as they expend all their energy fighting bacteria. So what the macrophages do is they kind of eat up the rest of the bacteria that are kicking around. But they also clean up the mess, right? So as the neutrophils die, they'll start to clean that stuff up. And then all the cell debris, all the little things that are left around, the macrophages are very good at dealing with that kind of stuff. And then there's a very important response that the macrophages and dendritic cells do, which we'll talk about in just a little bit. Okay, so part of this then is we have our white blood cells in the area. How does one recognize what a pathogen is? Like, how do they know, right? So there are two ways. One is using an opsonin. Remember that thing, right? So it's our proteins that coat the pathogen, and then our phagocytes grab the opsonin. So they don't even care. They see an opsonin, they're just going to grab whatever's attached to that opsonin, and then they're going to eat it. Okay, so that's one way our white blood cells recognize. The other is this thing called a PEP, pathogen associated molecular pattern, right? And it's just basically this generic term that refers to some sort of molecule that is only associated with a pathogen, right? So there are structures. If you, this is section 17.4. There's a really nice list in your book. I didn't want to put it in here because it's a big list. But it is a really nice list that you can look at just to get a general idea, right? But these are structures we've seen, right? So LPS, or lipopolypeptides, you know, only found in bacteria. Flagellin, right? These are proteins that make up the flagella of bacteria, only found in bacteria, right? Peptidoglycan, bacterial cell wall. You know, certain types of nucleic acids, certain types of things, right, that are present. All of these things are PEPs, again, very specifically associated only with bacteria, these various pathogens, and so they are then eaten up. They all have these receptors that are unique to the PEP, right? Collectively, they're called pattern recognition receptors, or PRRs, right? So PRRs are on our cells, our phagocytes, and they recognize PEPs. One of the most common PRR is called a toll-like receptor. I only put that in there because toll-like receptors are so vital to many, many cell functions. So you may run into this term periodically here and there, right? But basically they're receptors that bind to various patterns, right? Various things, and then they trigger some sort of response, right? So when things bind to these receptors, and they're often used to recognize viruses and bacteria and all these various things, and then stuff happens. And typically it's phagocytosis, or it could be cytokine release, and really it depends on what is being bound, right? Because it can be a viral structure as well, it can be a protozoan structure. So when it recognizes something, so you know, you can imagine how a virus binds to a toll-like receptor, right? The response then will be, okay, I need to secrete some interferons, right? Because eating it is not super important. It's also protecting the cells around us, right? Okay, but we have this pathogen that now got grabbed by this white blood cell. So imagine a neutrophil grabs onto a pathogen's PAMP using its PRR, right? Now it's going to eat it, right? So it does that. Again, we're going to keep this nice and simple. It's going to wrap the pathogen with a pseudopod. Pseudopod meaning false feet. It's actually like these extensions of the cell that it uses to kind of walk and crawl, right? But it engulfs it with this pseudopod, and it comes into this thing called a phagosome. right? And the phagosome is now this membrane-bound compartment. Very importantly, its pH is low. It has a pH of like 4 to 5. And then this phagosome, oh sorry, so it's got a low pH, right? At some point, the phagosome fuses with the lysosome. And remember, the lysosome is one of our organelles we talked about a long time ago that contains digestive enzymes, right? So all these enzymes in here that degrade things, this is one of the things that it does, right? So the lysosome then fuses with the phagosome to form this thing called a phagolysosome. And this is where our pathogen gets digested, right? So it doesn't get digested in the phagosome. It doesn't get digested in the lysosome. It gets digested when the phagosome fuses with the lysosome. And conveniently enough, the enzymes that are found in the lysosome are pH dependent. And guess what pH they like to work at? That's right, pH 4 to 5, right? So this actually prevents the enzymes from going haywire. If they accidentally get released from the lysosome and into the cell, right? The cell's pH is usually around 7, right? It's not going to destroy things inside the cell, right? It only will activate when it's in an acidic environment. Okay, so then our bacterium gets digested, and then anything that's undigestible gets excreted, right? So again, this is kind of like us. We eat something, we digest it, we poop it out, right? Interestingly enough, there are things called antigen-presenting cells, right? And we will learn more about this in the adaptive immune response, but they present a little bit of what they've eaten, right? You know, we get those kids that are like, they eat something, it's like, hey, you want to see what, or you like seafood? Here, seafood, and they show you what they're eating, right? It's kind of like the same thing, right? Except in this case, this antigen-presenting cell, of which macrophages and dendritic cells are vitally important, they will have eaten a bacterium and they say, hey, white blood cell, hey, you, come. I think you should check this out. Check out what I just ate. This doesn't belong, okay? And so they present this to parts of the adaptive immune system to help activate it. And we'll learn about that, probably more than you want to know, in Module 11. Okay, so the last section here I want to talk about is inflammation and fever, right? This is a very important part of our innate immune response. As we kind of coast to the end, we're almost done, I promise. Inflammation is a local response to infection, right? Remember, local, small area, right? And it's triggered in response to damage. So when cells get damaged, they will trigger the inflammation response. When they get stressed, they will trigger an inflammation response. When they're infection, they will trigger the inflammation response, right? The outcomes or the role for inflammation are threefold. We want to get rid of the pathogen, basically get rid of whatever's causing the inflammation. We want to remove damaged and dead cells, right? So during all this time, there's going to be a lot of stuff going on that we need to clean up. And then we want to initiate repair, right? Because we're going to have we have damaged cells, we have damaged tissue. We need to make sure we repair that. So inflammation actually is very important, right? We hate inflammation. We're like, oh, inflammation sucks. I hate it, right? But it's actually very important, right? However, excessive inflammation, as we will learn with any part of the immune system, right? We always talk about this Goldilocks zone, you know? This one has too much, this one has too little, right? We want it just right, right? Excessive inflammation results in severe tissue damage and it can lead to death. And we'll talk a little bit about excessive inflammation, excessive fever right at the end of the chapter. Great way to end our module. So let's talk about inflammation, right? So most inflammation is acute, remember, short and sweet, right? And the way this works is the inflammation response, things like histamine, things like leukotrienes, what they do is this process called vasodilation, right? It makes the blood vessels, the capillaries, bigger, right? Now, how does that help, right? So, you know, we think of like, I used to think of blood vessels as being like a big PVC tube, right? But it's actually not a big PVC tube. It's like a tube made up of a whole bunch of little pieces, right? Like a whole bunch of little Legos. So then when you swell, the Legos kind of stretch apart a little bit and you start to create holes between the cells, right? So this is illustrated real nicely here. We got these cells that make up our blood vessels and then it creates these little holes, right? So what this then allows for is this rush of everything that's in our blood vessels to the site of injury, right? This leads to a couple things, right? It leads to erythema, which is the redness because of all the blood in the area, right? So that's why inflammation looks all red, whole bunch of blood. It leads to edema, which is swelling, right? Because a whole bunch of extra fluid is in the area, right? And then the extra blood, the extra swelling is the feeling of the heat and the pain. And so that's why inflammation feels like the way it does, right? It's because you got all this extra blood in the area, right? However, with that comes the white blood cells, right, through diapedesis, as we talked about. And then with that also comes all these other white blood cells that can release various cytokines that can help fight pathogens, it can help clean up, and then ultimately help begin the tissue repair, right? So bringing all the good, and extra nutrients, extra oxygen, all these things lead to the side of inflammation, right? And so as much as it doesn't feel nice, it's vitally important, right? Because we have to bring this in here. And so if you don't have a nice inflammation response, this pathogen will allow to grow quite unchecked, and then we'll just end up having more troubles because we didn't get rid of it. It's sort of like, hey, you know, you can go through a little bit of a pain now, and then we can clean up our mess. Or you can have no pain, but then you know what? You're gonna have a bigger mess to deal with later, okay? So what happens if we don't clean up our mess, right? If we don't clean up our mess or are unable to clean up our mess, we have something called chronic inflammation, right? And so this happens when the acute inflammation was unable or just didn't clear the pathogen. Sometimes pathogens are able to like hide a little bit, right? And then we couldn't clear them out. Sometimes the product is not something that can be cleared. So, you know, if you have, let's say, rather than a pathogen that got stuck under your skin, maybe like a splinter or a piece of metal or something like that, you know, we're going to mount an immune response to that, our inflammation response, but we can't get rid of it because we can't eat it, right? Or the bacteria has various defenses to allow to hide, right? So what happens is we still go through our acute inflammation, right? And then our body says, okay, we're done, right? And then area heals, right? But it heals over top of what's causing the inflammation, right? But because we didn't get rid of what's causing the inflammation, you still got like this low level, right? Your body says, you know what? I'm pretty sure we clean everything up, right? But this constant this inflammation response. It's keep telling me to go and do inflammation, but we did it. So you get this low level of inflammation, very low level over long periods of time, right? And then what happens is because we are going to attract white blood cells, but there's not so much chemoattractant around, they move real slow and they all surround something. So why are we here? I'm not even really sure. But these chemicals, these cytokines want us to be here. And then so sometimes they surround this tissue that's releasing all these cytokines, right? And then so these are what are called granulomas, right? So these granulomas are just tissue that's surrounded by white blood cells, and it causes, again, just low-level inflammation over long periods of time. And over time, that can cause a lot of issues as it starts to cause damage to the tissue, okay? Okay, so in summary, inflammation is a local response to infection, local response to damage. The idea is that we want to get rid of what's causing it, i.e. the pathogen. We want to be able to clean up the debris, and then we want to repair tissue damage, right? Sometimes a local response is not enough. Sometimes our whole body needs help, right? So what's the opposite of local, right? So if you have a local response, the opposite of that would be systemic response. That's right? So fevers are a systemic response to infection. And fever generally is this overall increase in body temperature. And we've all had fever. We all understand that, right? And so the chemicals that trigger fever are called pyrogens. And the way that works is that in our brain, we have like what everyone always refers to as your thermostat. And the pyrogens are chemicals that reset your thermostat, right? It just says, okay, let's turn it up a little bit. And we can have endogenous pyrogens. We can have exogenous pyrogens, right? Exogenous would be things like LPS, for example, right? Which are, remember, the endotoxins, right? That are from gram-negative bacteria. Or we can have, which then will trigger leukocytes to trigger these various interleukin-1, interleukin-6, interferon gamma, tubular necrosis factors, all these various endogenous pyrogens, which then goes to this pathway to our brain and it says, okay, time for fever. And then we simply increase body temperature, right? Now, we always think of fever as bad, but like with inflammation, fever is good, right? Because it enhances the innate immune response, right? It stimulates leukocytes to kill pathogens. It allows them to work just a little bit faster, much better. Pathogens generally grow best at like 35, right? So by increasing the temperature just a couple of degrees, it actually slows their growth down just a tad, right? And what it can also do is it can limit iron uptake by pathogen, right? The sequestering of iron, right? So it's again, very important nutrient for pathogens, right? By increasing the body temperature, we make the available iron limited to pathogen. Okay, so how does fever feel like? You know, and we always get people who know what fever is, but they don't really understand fever, saying stuff like, oh, I feel really warm, I must have fever. Well, that's not really what happens, right? When you have fever, the response is going to be to take all the blood that's circulating around your body and to bring it into your inner core, all right? So then that has a couple of effects. First of all, right, all the blood comes in, right? Which then moves the blood. Remember, the blood is the warm part away from our skin, right? And so that's where we are. Our skin is going to start to feel clammy and cold, right? And then you get the chills, right? And the chills are the shivering, right? So remember, what do you do when you're cold? You shiver, right? And the shivering, the muscles twitching, warms you up, right? So how's the body going to get you to increase your temperature? Well, it's going to make you shiver like crazy, right? So actually, when you get fever, right, if you are saying, oh, I feel so hot, you don't have fever, I promise you, right? When you start shivering and you feel cold, that's when you have fever, right? So you feel cold, like, oh my goodness, I feel so cold. Because remember, your thermostat has been reset. So now your normal temperature is no longer your normal temperature. It's too cold. So you start to feel cold, right? And all that shivering starts to increase your body temperature, right? And these are what's called the chills. So the chills is the sign of the onset of fever, right? Then our body temperature raises to kind of that area that it wants, and then you kind of feel like doo-doo for a long time, right? Finally, infection has been cleared, right? The crisis phase is the end of fever. So now what happens? Well, now we want to move the blood back to the surface, right? Bring it close to the surface to let the heat leave through your skin. Right? What do you do when you're hot? You start to sweat. So you start to sweat, and so that's when you feel like your fever is broken, right? So the crisis is when your fever is broken, right? And so typically the end of the fever is seen by the sweating. And you know, I think we've all done this, right? You go to bed feeling sick and you wake up in this pool of sweat, right? And that just means your body has finally broken its fever, right? And there you are, just all like trying to regulate back to normal temperature. Okay? So that's fever, right? And again, overall helps your immune system work better. Again, we don't like inflammation. We don't like fever, right? But inflammation, fever are vital for the immune response. So important. Or in fact, inadequate inflammation, inadequate fever is, really allows your pathogen to overwhelm the body, right? And then we'll talk later about the power of the adaptive immune response. These are the knights of the immune response. They go out there and they fight all the bad guys, right? However, as powerful as the adaptive immune response is, it is better to have an innate immune response, right? Because the innate immune response prevents you from really getting sick. Our adaptive immune response only works when you are sick, okay? So with that said then, if the adaptive immune response, which is slow, right? If we're solely dependent on the adaptive immune response, you will probably die before it's ready to go, right? So that's where the innate immune response is so important. So inflammation, fever, very important for all these things, getting rid of the pathogen, right? Eliminating or cleaning up and then repairing the tissue, right? However, excessive inflammation is bad, right? Because excessive inflammation and fever cause major tissue damage. And so this leads to this phenomenon called a cytokine storm, right? This storm of cytokines as all these cells are just releasing cytokines like mad, right? And again, a little bit okay, cytokines are important, right? But this constant release of cytokines everywhere leads to this excessive inflammation, excessive fever. And cytokine storms create these life-threatening conditions, right? In fact, many diseases are fatal not because of the pathogen, but because of the cytokine storm. And a really good example of this is SARS-CoV-2, right? The death from COVID-19 is not because of the virus, right? The virus does its thing. It's going to go in there. You're going to feel like doo-doo. You know, you're going to feel like a cold, right? Sometimes a wimpy cold, or sorry, flu. Sometimes like extreme flu, right? But that's not the part that kills you. What kills you is the cytokine storm phase. After the viral phase is gone, after our body has cleared the virus, there's supposed to be this response that says, okay, immune system, you have done your job. You can now go back to rest. But there's sometimes this tendency for the immune system to keep going. And no one is really sure why it happens because of SARS-CoV-2, but it does, right? And so you get this crazy release of cytokines that they just won't stop, right? And then it's that phase that's the dangerous part. That's the phase that leads to the life-threatening death associated with COVID-19. We do know that things like superantigens, which is one of our endotoxins, or sorry, one of our toxins we talked about, not the endotoxins, exotoxins, we talked about in section 15.3, module 8, I believe, maybe 9, that where, you know, superantigens trigger cytokine storms, right? And they do that because, you know, causes a lot of tissue damage. A lot of tissue damage is good for the pathogen. And then some infections, like I just talked about, can trigger cytokine storms, right? So some infections, just for whatever reason, prevent our immune system from shutting off properly. Okay, kind of this is not the fun part to end with, right? So this is like this doom, our immune system, right? But the innate immune system, so important in helping us prevent some serious infection, right? And then really help bridge the gap, give time for the adaptive immune response to mount, which takes a long time, 4 or 5 days to really get a good response going, right? So the innate immune response, very important in giving time for adaptive immune response to come, because sometimes the innate immune response is not enough. So our adaptive immune response needs to come. So we're going to see all about that in module 11. Until then, good luck on your quizzes, and I will see you soon.